All right, so in this final lecture for the bootcamp, I want to go into some concepts used in pathway enrichment analysis to help you guys plan such an analysis for your own experiment. So, the reason we want to do such an analysis is to make sense of gene lists. So, after the entire process that you just went through of mapping your sequence reads, quantifying them for each gene, and discovering the expression level in each of your samples, you have this full set of gene expression data, which you even took further and performed, for example, a differential expression analysis, and this reduced the list of 15,000 expressed genes to, let's say, a group of 400 genes for which you saw an upregulation in your experiment with respect to your controls. And now you still have quite a large list of 400 genes to try to understand what's going on in your samples. And you can do an old school sort of analysis where you go through each of these genes and you try to make sense of them together and figure out what is a possible mechanism acting in your experimental condition or any other shared feature of these 400 upregulated genes. But a pathway analysis might lead you to that information in a much less labor-intensive way. And another example is the one I showed you guys on the first session, where not only do we know which genes are upregulated or downregulated, but for example, by performing a clustering analysis, we can subdivide them into groups which show equal temporal expression. So we have, for example, this labeled cluster here where genes are induced very quickly after our LPS stimulation and reach their maximum expression at 1 hour post LPS induction. So this is the same case where pathway analysis can help you identify commonalities between the genes that have been assigned to each of these clusters. And there are many examples of pathway and gene set databases that contain the information you need for this kind of analysis. And I've put some of them here for your knowledge. For example, gene ontology, which I'll go into in more detail, and the Panther classification, which is somewhat tied to gene ontology. Reactome, KEGG, and WikiPathways are all databases of known biological pathways which are well described. And there's also this molecular signatures database from the Broad Institute, which can be extremely helpful if you're dealing with human diseases. And again, we want to ask, using these databases, what biological processes are altered in your experimental condition. It can lead you to a better interpretation of your results and even suggest a potential mechanism that is commonly impacting the RNA levels of many genes at the same time. And other examples of lists beyond what I've put here are, for example, all of the possible targets of a microRNA or all of the regulation targets of a particular transcription factor. You can compare your results to lists of mRNAs that interact with the protein of interest, or even protein-protein interaction lists obtained through immunoprecipitation assays. And you can compare your gene list, for example, the expression of the same genes in other tissues, or any kind of literature curated list, and any list of genes associated to a particular disease. So let's go into gene ontology a bit further. An ontology is just a structured system for describing knowledge. And what gene ontology tries to do is to associate gene products to three different categories. And these categories are: first, cellular component. So Go terms that include genes in this category describe, for example, where those products function. For example, in the nucleus or in a particular organelle. Or they describe associated genes that are components of a particular cellular machinery. For example, genes that are components of the ribosome or proteasome, or take part in the formation of any kind of protein dimer. The second category for Go terms is biological process, and this one describes organized assemblies of molecular functions. So these are not quite pathways. For example, they can be very broad, such as cellular physiological process, or they can be very specific and include only genes related to alpha glucoside transport. And you have a third category that is molecular function, and these include terms that are related to activities at the molecular level. Again, they can be very broad, such as catalytic activity or binding activity, which would include transcription factors as well as RNA binding proteins. Or they can have also narrower terms, including toll receptor binding, for example, which would be relevant for the example I gave you of the LPS stimulation experiment. And the difference between a biological process and a molecular function is that the biological process must have more than one distinct step. So while the molecular function can be a single action, the biological process must be two or more. So let's say, for example, that we did choose the gene ontology database, and we want to evaluate if there are any pathways that are upregulate, over or underrepresented in a particular gene set of our interest. And in this case, we'll go back to that example where you guys just computed the differential expression and you have your results for all of those genes, and you established a cutoff where you decided that genes that meet that threshold were either upregulated or downregulated in your samples of interest. So after establishing such a cutoff, we were left with 435 genes which we considered induced in your experiment compared to the control. And now we examine what this list of genes has in common with a particular Go term of our interest. So this includes, for example, 800 genes. And you realize that the overlap between your list and this Go term means that 50 genes that belong to this particular pathway or cellular component are also present in your list of genes that were upregulated. And the question you want to ask now, is this surprising? So how do we define if this overlap of 50 genes is what you would expect by chance, or if for some reason there is an overrepresentation, so there are more genes than we would expect by chance that belong to this particular term upregulated in our experimental condition? And to answer such a question, I want to first introduce this very cool experiment that I've just recently heard about, where they determined that these Kia parrots from Australia actually understand probabilities. So, what they do with these parrots is they train them to understand that only the black tokens will be exchanged for food. So, the parrot knows that an orange token is meaningless to its feeding time. And then the researchers present the parrots with two jars composed of different quantities of black and orange tokens. And the researcher chooses one token from each jar, and all of this with the parrot watching, and it shows the parrot and it lets the parrot choose which hand does it prefer. So the hand that went into the jar with more black tokens, or the hand that went into the jar with more orange tokens, but still has some black tokens in there. And I really encourage you to go watch the video. It's a 4-minute video and it's a really cool explanation. These parrots actually know that they have a higher probability of getting a black token if they choose the hand that went into the jar containing more black tokens. So they understand the likelihood of getting a positive response. So these parrots can actually answer your question of overrepresentation of a particular gene set in your differential expression analysis, because this is the same idea. You have a group of genes and you have genes that belong to a particular Go term and genes that do not, and you want to evaluate the probability of reaching into a jar and obtaining a certain number of genes that belong to your category of interest. So let's go into this a bit further. How do we determine if this overlap we observe is significant? If we put numbers on this example now, you have your universe of genes, or your jar, where you determined 15,403 were expressed, and you also have the knowledge from the database that 800 genes in this Go term, out of the 800 genes in this Go term, 500 of them are expressed in your particular sample of interest. And that means the rest of them are not present in the ontology. You also made a selection of 435 genes, shown here in this red circle, that were detected as upregulated in your differential expression analysis. And in those 435, 50 of them belong to your particular Go term. So how can we assess this enrichment using probability? We can use two types of tests, which are, in this case, essentially the same thing. So either the hypergeometric test or the Fisher's exact test will give you the same answer. The hypergeometric test is exactly about the chances that sampling from this universe of genes, a group of 435, what is my probability of obtaining 50 or more genes? So in this case, you can use R, and this is how you would input the same numbers I just showed you here into the R function p hyper. And the resulting p-value tells you the probability of getting more than 49 hits, so 50 or more, from this gene list in your sampled 435. And you can see that by chance alone, it would be an extremely low probability. So this means that this particular Go term is overrepresented in your gene set, and it might tell you something about the biology of your experiment. And for the Fisher exact test, in this case, it's exactly the same as the hypergeometric, and it'll give you the same p-value. But in the case of Fisher exact test, you need to compute a contingency matrix, which can be a little confusing to write up correctly. But essentially what the matrix is 2 rows and 2 columns, which will tell you exactly these numbers. So for example, how many genes are upregulated and also part of your Go term, or not upregulated but still part of your Go term. And in the second row, how many genes are upregulated but not part of your Go term, and neither upregulated nor part of your Go term. And once you write this matrix, you can input it into the function as you see here, and you essentially get the same p-value. And the last thing that I want to mention is that for this kind of analysis, it really matters the genes you choose as your background and test. For example, Albert touched on this a little bit when he was introducing you to Go analysis using the browser. And what it means is the background set can be either the total set of genes you quantified. So for example, before you did any filtering on gene expression, you had from your RNA-seq analysis a table containing 30,739 genes. And before doing any filtering, you also have 800 genes in total that are associated to your Go term of interest. But if you consider only the genes expressed in your sample, then you reduce this list to 15,000, and you reduce the second list of the Go terms to only the expressed genes in that term, which are 500. And this can significantly impact the probability you're calculating for observing that overlap. So think about it in terms of the jar of tokens again. If you have black tokens in the proportion of 800 out of 30,000 in a jar, it's a different probability of obtaining one than when you have 500 tokens in a jar composed of 15,000 tokens. And this is just to show you an example of the same hypergeometric test. If you run it with the numbers for the filter gene list, which we did before, which is the proper background to detect any changes that happened due to your experimental conditions, as opposed to using the incorrect background of the full universe of genes, which completely changes your probabilities and will give you an inflated p-value telling you that it's more unlikely that you've obtained this 50 gene overlap, but it's just artificially inflating that due to your larger background. Using the incorrect background will also lead to other spurious events. For example, if you consider the gene expression profile differences in different tissues, and think about, in this case, we're dealing with liver, and we've determined that there's 15,000 genes that are expressed in this particular tissue. This means that there's a higher chance that any impacted pathway will be related to specific liver functions. So using the incorrect background of the full gene set will lead to the identification of several pathways as significantly enriched just because they're more likely to be expressed in liver cells. And by using the proper background of the genes that are actually expressed in your sample, you control for that and you don't waste time with enrichments that are simply due to a gene expression profile in that tissue. So assuming you got many different enriched pathways, I just want to remind you not to forget about the false positives that we discussed when doing differential expression analysis. This is the same case where we have to correct for multiple testing. So with the gene ontology database, for example, the current release includes 44,508 Go terms, and that is how many tests you're performing if you're comparing your one gene set to each of these pathways to detect any significant overlap. So don't forget that you should always do your thresholds on the adjusted p-values to avoid chasing false positives. This is just to show you an example of gene ontology analysis done through the Panther online tool using the upregulated genes in that example experiment we've been working with. So these are all Go terms from the category biological process, and in a tool such as this, and you can use different ones, including the version in the browser. You'll obtain a table giving you the FDR adjusted p-values for each particular Go term, how many genes were present in your list and how many genes were present in your background reference that belong to this pathway, what is the fraction of genes you would expect, and what is the fold enrichment in your list compared to the expected. And there is one final approach that you can take to evaluate gene set enrichments in your data, and that is called the GSEA. It stands for Gene Set Enrichment Analysis, and the advantage or the main difference from the overlap tests we were performing before is that it's rank-based. So there's no cutoff required. You don't need to predefine your list of upregulated or downregulated genes. So what you're doing in this case is taking your full list from differential expression results and ordering those genes by their fold change. So any gene that was tested in your DE analysis will be in this list if they were significant or not in terms of their expression change from experiment to control. So in this 15,000 gene list, ranked by highest fold change in your experimental condition to lowest fold change, you're going to look for genes that belong to a particular Go term, and if they are skewed in their distribution of your ranked list. So is there an overrepresentation of genes that belong to this Go term in genes that tend to be induced in your experimental condition or downregulated? And here's just to illustrate an actual result from an analysis I did with excitatory neurons, where you can see that the majority of genes in this particular pathway I'm testing, and they're shown here with black lines, tend to be downregulated in these cells. So this is just a graphical representation of this significant skew, but there will also be a table result where you can select on a p-value threshold, or even better on an FDR threshold, if you are performing multiple tests of the gene set enrichment analysis as well. And I'll leave you with some references for the gene ontology homepage, as well as the GSEA documentation, which is really comprehensive, and this nice article that has good information for good practices and other tools you can use for pathway enrichment analysis.